Hi everyone, it's a pleasure to be here, and I want to thank the organizing committee for inviting me to the summer school to share with you some of our experience in discovering innovative therapeutics. I thought today that what I'm going to do, I'm going to take you slowly into drug discovery and really how we can discover innovative drug, and then we'll go with some few examples. But while I'm doing that, I'm going to share with you what the challenges of doing drug discovery, because it's easier to make compounds and it's easier to go and buy them made. But it's very important that you treat the disease that you make the compound for. So what I thought actually early on, just to say in a simplistic term, because many people get confused when we talk about drug discovery. Yes, indeed, our first task is to treat diseases. And so you take some ideas, make drugs to treat the diseases, and this could be done by pharma, academic institution, and others. But believe me, I've been working with different countries. In order to do biomedical research, you really need to have a good foundation of drug discovery, because it's an integral part of doing translational research and others. So, but in simplistic terms, yes, indeed. So when you want to treat a disease, you want to know first what caused the disease or what's really being implied. And that's why we call it the target. Like, for example, when we work in antidepressant, if you have two chemicals in the brain, you have less amount, like serotonin, norepinephrine, you will get depressed. So for me as a medicinal chemist, how I can raise the level of these two chemicals in your brain. And that's what we call the target is the neurochemicals. And then what we do, we'll get molecules, or what we call, refer to them as maybe some of the hits. And then we take these hits and do like cosmetic surgery, try to optimize them to make sure they're effective and actually work in animals so you can take them to human. And that's the process where actually usually refer to like you have a lock and you would like to make the proper key to unlock the lock. So let me just say simplistic, we talk about targets. You think there's so many drugs discovered. There are not too many. It's about 1,400 drugs has been approved by FDA. But they work on about less than 350 targets. So when people thinking about all this disease and company every year putting drugs, they really don't too many targets there to work on. But when we got the human genome back on the last decade, and people can argue how many genes in the human body, you hear some literature say 70,000, 100,000, but mostly agree on around the 30,000 genes. So that means you can look at the protein being produced and some of those targets, which could be the future drugs for treatment, any diseases like we saw in Yana's slides, like maybe pancreatic cancer or Alzheimer, where there are no drugs for them. So now we have all these new targets, almost 5,000 targets. But the trick here, how you correlate these targets to the disease. And that's what we refer to as target validation. So most of the pharmaceutical companies spend a lot of money and effort to do target validation. And that's where drugs fail, because maybe you don't have the right targets to work on. When we work in discovering new drugs, these drugs could be small molecule like chemicals, you take the pills, or could be injectable, biologic, or could be vaccine. And all these three platforms follow the same process I showed you, from the target going to have an end product. So really, when you discuss these, for my expertise as a small molecule or chemicals, but we run also some of the small molecule with the biologic, like the antibody, I'm going to share with you, and Yanis referred to in one of his slides, where he have the antibody conjugate. So we'll come to that. Here is an example, for example, of some of the platform. You have small molecule, the drugs we know, or biologic, for injectable for inflammation, or for vaccine for pneumococcal, or when the infant get born, you give him all the series of vaccination and so on. To discover whether it's biologic or small molecule, it's a really lengthy process. And doing drug discovery, as we're talking here among ourselves, this is just this part. But you have to go through the whole development process. And this process is tedious because you need thousands of molecules to pick the right molecule to put it in man. Because if I go to the clinic, I want to make sure what I'm putting in human is effective and safe. So here, from all this, you get only maybe a handful, go to clinical trial, and maybe you get one marketed product. And that shows you how challenging it is. So there is a lot of failure rate. And all this costs money. And if you want to talk about drugs costing money, what we do is really not, it's expensive, it's cheap. Here you're talking about when you go to phase 3, 10 times. 10 times the money is spent just to stick it in human and to see if it has an effect. But what we're doing from ideas to get the molecule is really a fraction of the cost. So for me as a scientist, I say, if I put something in the clinic and goes all the way and then it failed, that means it failed very costly. So for me as a scientist, I tell the company, let's try to fail here rather than fail later. So fail early and cheaply. And that's what takes me to you to discuss with you what we call attrition rate. You see that funnel, you put so many in the clinic, but what comes out of it is only a handful of molecules. So some of these molecules is dying for some reason here. And here are the reasons. I've shared them with you. There are 3 major reasons here. The drug doesn't produce the effect. If I'm treating an ulcer and I don't treat the ulcer, there's no efficacy. Or the drug, which really want to treat something in the brain, does not go to the brain because the physical chemical properties are not the right physical chemical properties. Or the drug, after the human take it, produce another effect, off-target effect, toxicity. So how we can treat this early on? So for us as medicinal chemists, we realize that we can reduce the failure rate because of the physical chemical properties tremendously. So as a medicinal chemist, we succeeded in that. Then you take this molecule now, which can reach the brain and others, and look for the other parameter. But the major, here. For drug, when you take the drug to give it to human, and the drug doesn't work, then one would ask himself so many questions. I list them on the slide here for you. Well, somebody would say, you selected the wrong target. So if you're going to treat something and I pick the wrong chemicals in the brain, the drug is not going to work. Or some people will say the in vitro pharmacology really doesn't look good. Yes, indeed, because there is the mechanism, is poor mechanistic approach. Or as you see in the rest here, all these reasons where you can improve in your medicinal chemistry approaches. And how we improve in medicinal chemistry approaches, when I was hired many years ago, when we discovered Effexor, and you would be surprised, I showed you 1,000 compounds to get a drug. My team made 60 compounds. And out of those 60 compounds came a drug which we sold $4.5 billion. But that was then. Now it's a different ball game, because in the old days, you can say just put anything in the clinic, and then one of them will make it. And that's actually nowadays is not a good approach, because you really have to put your best foot forward. So you want to make sure the molecule you put in the clinic going to reach the target and going to produce effect in animal, and all these things. For us as medicinal chemists, we refer to this as multidimensional lead optimization. I'm not going to optimize just the biological properties, so the biologist will say it's 2 nanomolar. It's not good enough. I want to optimize solubility. I want to optimize the druggability of the molecule. And that's what we refer to multidimensional lead optimization. The drug discovery process, as I said, as a medicinal chemist, is really like a juggling act. You make molecules, it's very high molecular weight. How I reduce them? You make molecules not soluble. How am I going to make it soluble? So this is actually a juggling act. We work on that. So when I was heading chemistry many years ago, I decided that we can do these two optimization that if I get a molecule which bind very well, that's not a drug. It's a good ligand. If I get a molecule very soluble, it's a good physical chemical property. It's not a drug. We want to combine these together, and that's where you get a good drug candidate. So what we did, we established a group there where when they do the optimization, they have to keep in mind what the important phases of drug action. To get the pill, you want to make sure it's well absorbed. But don't forget, you give the drug to the body, it make an effect on the body, but the body make effect on the drug. It want to get rid of it, they metabolize it, excrete it. So we want to make sure that for our process, that the pharmacokinetic is good, the physical chemical properties is good, then you want to make sure you made the right compound. So basically, we have two objectives: optimize activity and optimize property. And we do those in parallel. We do both these activity in parallel. We have a set of assays, and we can do this automated assay, where you make compounds and you give it to an analytical chemist. And he will take this assay and give you solubility and all that. Or with the biology, give you the biological activity. So the chemists now do these two tasks in order to be successful. And one of the things we've done, we built this ensemble of assays, and we built those at Temple University. So when we make a molecule, we know that this molecule will be predictable for biological activity by reaching the target, absorbed, orally bioavailable. So for us working on the bench as a medicinal chemist, we can design the best molecule. There is a lot of tools available, computational tool, screening tools and all this. So what you do here, you can use for finding a good lead, high throughput screening, you can do virtual screening, fragment-based screening, all that. Then you do the optimization actually along the way to get your molecule. So let me take you for a few examples here to show you, for example, virtual screening. Janos talked a little bit about some steroids and so on. When we were working to come up with progestin non-steroidal antagonist, there are no X-ray structure for this, for the antagonist, but there are for the agonist. So what we can do, we can actually build the model by taking these helix 12 and push it away from the agonist. Then we do screening and we come up with some leads. And that's what we came across from the library, because we have built an homology model. Then the chemist will take that and try to optimize. This here is just 5 nanomolar, and then we remove the bad functionality, because most of the drug, when you have guanidine, urea, sometimes you want to get rid of these, and that's what we did, and this molecule moved into the clinic. I want to share with you another example. If you work in CNS, there are not X-ray structure for many targets in CNS. So you actually build your pharmacophore. So when we work for treatment of antipsychotic or schizophrenia, what we try to do here, we know from the literature, there are some molecules which built in the dopamine structure inside. So we took the modeling, the Confa model, and we did that, and we tried to build on some of these molecules the dopamine structure. And then we tried to do screening around, and we built around that actually many molecules, and that came out of that. The molecule advanced to phase 2 called Aplendo. And that came without having X-ray structure, but using some computational tool and building the pharmacophore. Let me give you a little bit about when you have the structure, and that's the chemist's dream. When you have a structure of the protein, that means your success rate will be actually more enhanced. So when we work for inflammation, and we know certain enzyme in the body like the TNF alpha, tissue converting enzyme, and that release the TNF alpha, which can form rheumatoid arthritis. So for me as a chemist, I say how I can treat that disease. Two things. If I can get rid of TNF alpha, then I treat the disease. How you get rid of that? Well, you inhibit the enzyme which causes it. This one thing. Or you do biologic, you get a receptor, can suck it as a sponge. And that's what Embryl all about. It's a soluble receptor from the protein. So for my group to work on that, we said, well, let's design actually an inhibitor for TACE. And that's where we found that you look at the TACE, unlike the other MMPs, matrix metalloproteinase, the TACE has structure like this, but also have this addition, which is 90-degree angle, which people call it sometimes who play golf, the dog leg to the right and so on. So for me to tell the chemist, I said how we can fill this angle to come up with selective mode, because we know the structure. So what we did here is the TACE, you can see that, and the molecules, and here is the extra angle of TACE, different from MMPs. So the chemist worked with, said, well, this is the lead molecule. How I can fill this part? So what he did, he put acetylene ring. See that? And that acetylene ring filled this groove here. And look at the activity. Tremendous selectivity between the two enzymes, closely related. So when you have an X-ray structure, you can design new molecules. Let me take you now for one of the last examples in the first part of my talk. As we said, when you get these analytical methods I showed you, the molecule is stable, the molecule is soluble, how this can help you to build the novel drug candidate. Well, I'll show you here an example for this here anti-cancer drug. When we did high throughput screening, we came up with these compounds. And what I do in my group, we rank the activity, physical chemical properties with color. Red is no good, same when you drive your car. Yellow, okay. Green, we want them all to be green. So what we do for the chemist, we get these data for them, and we say, look, this molecule here is really needs some optimization, is particularly in the stability, because the CYP activity is actually very low, the stability for this. So the chemist agreed that you have a labile group here, and I cut the story short by telling you that they actually put this alkoxyamino, and you look at the CYP stability was high, the molecule was more potent, and moved from the red to the green, and that molecule actually moved to the clinic. And we made it large scale for the chemist in the audience. Just, we have to say something to show you that 5-step synthesis from commercially available starting material, we were able to make enough material to move into IND filing, and actually after that moved into phase 2. I hope that I just tried to show you some of the challenges facing attrition, because not every compound you make will succeed and make it to market. But at least you can do your part. You put forward a molecule which is efficacious in biology, and it's good drugability. Drugability means stable, absorb, and it can reach the target. And I showed you an example with the impact. The one I showed you where we moved all from the red to the green, or the others. So now let me take you to some real example like what Tiana's did, and talk about some innovative therapeutics. And these are the compounds I'm going to talk about, which I was pleased that it came from my group. And I'll share with you some of the thoughts, how this came about, and how we discovered antidepressant drug, Effexor and Prestige. So many of you know some of the family member or friends or so who suffer from depression, because we all know that in the current days, I mean, we all stressed out. And I'm talking about depression, clinical depression, not depression where you lose your dog or you lose a friend and you get upset. That's not a clinical depression. And that's why there are a lot of definitions for treatment of depression. So it's highly prevalent. Unfortunately, women suffer it twice as men. I guess men frustrate the women, so that's why. So, and there's a lot of people actually is severely impacted, especially because when you know what the symptoms are, there are two major symptoms for depression, emotional and physical. And usually the physical part, where you seek advice, you go to the doctor or you need medicine. Why? You get some wives, she doesn't care with the children, she doesn't want to go to the kitchen, they don't want to work. So when you take the medication, it really tremendously improvement in their physical symptoms and so on. So the old therapy, and I summarize them here for you, they're not really that efficacious from the monoamine oxidase inhibitors and tricyclic antidepressant and so on. So that led pharmaceutical company to focus on how we can treat depression. Well, it's very simple. When you take your pill for depression, it doesn't work that quickly. You need 3 weeks or 4 weeks. So you need rapid onset of action. You need to minimize side effects. So when we all embarked on that, we all realized that the biogenic amine in the brain are really responsible for our mood and memory and so on. Particularly when you look at the serotonin and norepinephrine. And that's where we realized that reduction in the serotonin and norepinephrine associated with depression. And then one would say, okay, it's easy now, we can raise the level of these neurotransmitters. Indeed, some company decided we're going to focus on raising serotonin level, and here is drug discovery. Other companies said, we'll raise the norepinephrine level, and here's the drug discovery. So when I worked on that project, I said, well, in order to be rapid onset of action, why don't we raise both levels in one molecule? And that was the approach, where we said we're going to have norepinephrine serotonin reuptake inhibitor, or it's known as SNRI. And that's actually was done first by Wyatt a few years back. So the idea came about, and I told you, you need to have good starting point. That's why high throughput screening, when somebody asks you, why you have high throughput screening? Because you have to start somewhere. And so every company have a bank of molecules, and they screen to get a lead. Then you can go from there. So for me, I knew that the company Wyatt had a drug in the clinic for analgesic called ceramidol, and that's the drug. So by looking at this without doing any computation, I said, it has the element of serotonin and norepinephrine structure, but the drug here has 3 chiral center. That means the molecule here, you have so many isomers. So let's try at least to move the hydroxyl group from here to here. By doing that, you have only 1 chiral center. And we did this. Then we tried to build in the serotonin and norepinephrine, and at that time we realized that having this group here in gamma-fexin has a little bit activity as antidepressant. So we moved from analgesic to antidepressant, then we did the chemistry, and we came up with the drug, which called Effexor. And that drug here, to our surprise, even though it was 3-step synthesis, was very intriguing in its biology. And that's actually where we reached to, without going through detail of biology, the one advantage we found that patients who take serotonin reuptake inhibitor, like Prozac, and they don't respond, when you give them the combined with serotonin and norepinephrine, they respond. And so that drug became actually a clinical success. But of course, there is nothing in this, day and age without surprises. The drug have a drawback. And what we say, the drug actually, when you take it, as I said, the human affect the drug. So what happened here, that drug being metabolized in the liver, and that enzyme here, CYP2D6, will take the drug, remove the methyl group, and you get these metabolites here. But in that case, some patients, some of us, have a lot of 2D6. So those will be extensive metabolizer. And some have low level, will be poor metabolizer. So how this affect your efficacy? Indeed, because look here, when you take the drug, it goes all the way down in intensive, but if you have the metabolite, the levels stay the same. So we went there and we did the study. We convinced the company that this metabolite in its own is better than Effexor. And indeed, this methyl, which is without the methyl group, became the drug, which is Prestige. And that actually now, you take one dose. With Effexor, you have to titrate the patient. You give them 75, 100, 200, because you don't know how they're going to metabolize it. Here you give them 50 milligram, one dose fits all. And that's actually was. So other company, when they realized that, they said, let's do the same. They went and did other drugs here, but as we all know, they are no efficacy like Prestige and Effexor. Actually, some of them became more into pain than depression and so on. So that shows actually how sometimes when you do first in class, you bring actually more innovative therapeutics. Let me take you now to another area which is close to my heart. Actually, we were working with natural product, and not too many companies work in natural product. They phased natural product back in the 80s and 90s for simple reason. Why work with natural product and they have high throughput screening? Because you have the chemicals, you get the lead. In natural product, you need to identify and you need to optimize on large scale. So many companies, but in our group actually work with me. We were very fortunate to have talented group, and they were actually able to do this in a relatively short period of time. So the strategy is, you take the natural product and you make it a drug by itself. Or you take the natural product and you do simple chemistry, semi-synthetic, and you make a drug. Or, like what Yana showed, you take the natural product and stick the antibody and you make a drug. Or you can feed the microbes and do some feeding experiment and you do some changes in the natural product and you can reach a new drug. And that's actually was the approach we used, and we were actually successful in generating many of the natural product candidates. Let me share with you one of the flagship projects we worked on. It's called the rapamycin. That natural product was isolated from the Eastern Island, South Pacific. And what we used to do, we send some of our natural product chemists to different countries, come here to Brazil or others, and you get us with us some soil sample. So we have soil sample from different countries. Then we do fermentation, and then we put them through screening for cell line to see if they're anti-cancer or not. Believe it or not, it was easier in the old days. Now countries prevent people from taking soils with them banned. You have to get a permission, and we've been doing that, getting soil sample everywhere. So we got this soil sample and we identified this molecule, and that molecule, just to cut the story short, we realized was immune suppressant and was good for treatment of transplantation rejection. So if people go for kidney transplant, they can use this drug. But more important for us as a scientist, the biological activity of this molecule is not just work with the FK binding protein. No, we discovered another kinase called mammalian target of rapamycin, or M2. And that actually showed that this side of the molecule bind to this, and this side of the molecule bind to the other protein. So what we did, we explored the activity in animal, and we realized because of the M2 with the binding protein, you can have multiple activity. And that was a task. We said we can get drugs in all this area. Indeed, that's what the team did, and they took the rapamycin, the 31-member ring, all this pharmacophore, it has 15 actually chiral center, and we put a whole bunch of chemists to work on that. And indeed, that's what we decided. Particularly, Jerry Skotnicki in my group decided that he's going to focus here for one reason. It's far away from the binding protein. So he said we can do C42 modification. And they worked very effective on that. And you can see all the activity here from different molecules being made. Almost 700 analogs with 100 patents is made in this compound. And that's actually was the strategy we approached in order to reach several molecules to treat several diseases. And here let me focus on one disease in cancer. And that's what Jerry did, and you will see the molecule is really very simplistic. You take the rapamycin, and he put the hindered ester. Remember I told you the 42 modification? And that's what Jerry did. And that hindered ester, when we screened that in cell line, we were amazed that how activity was shown for several cell line in cancer, like breast cancer and ovarian and others. And what most intriguing, that we realize it also in human glioma, and you don't get too many molecules. You think it does not cross the blood-brain barrier. And so we convinced senior management that let's put this in the clinic, and we put this in the clinic. And here you saw the drug, which is called Toricil versus interferon. And I'll give you a little bit secret for how pharmaceutical companies work nowadays. If you look at all the pharmaceutical companies, they all work in one area, all of them, which is cancer. One will ask himself, why do they work in cancer? Because it's the easiest drug to be approved. I'll prove it to you. Here, you see, Toricil increased the lifespan by 3 months. If you prolong life of patient against other drug by 3 months, FDA will love you, will give you approval. And that's why, unlike when you treat Alzheimer's disease and other. So when somebody tell you, we are really working in cancer because innovative, it's not true. Because it's easy to get drug approved for cancer by looking at the prolongation of life. So this is another thing. So that drug was approved for renal cell carcinoma and was launched in 2007. Okay, let me take you from the same molecule, how you can divine a new molecule for stroke. And everybody will think that's such a gigantic molecule. What do you mean for stroke? Well, it's a very simple reason. All these drugs you see here, they were put in the clinic for stroke. Over 1,100 drugs. They all failed, miserably. There is no drug succeeded for treatment of stroke. And the reason is because all of them hang on neuroprotection. If your brain cell had a stroke, then mean the brain cell died, how I can protect them from dying. That's not the answer. So I think people thought about that, how you can make this brain cell regenerate. So there are other factors, not just neuroprotection, but neuroregeneration. And that's where we decided how we're going to approach, because we were actually observant of seeing some of these immunophilin. They are not just being neuroprotectant for this animal model, but also they actually increase neurite outgrowth and promote neuronal survival. So we realized that some of the immunophilin, they are also neuroregenerative. And we tried to find the reason how they do that. So we tried to check for FKF binding protein, some of these, but we realized that the compounds, rapamycin, which have mTOR activity, is less than the molecule which does not have mTOR activity. So we realized that maybe the mTOR is not that effective for neuroregeneration. How we can separate immune suppression from neuroregeneration, and that was actually our task to do this. I showed you chemistry here. So chemist Ed Graziani in my group came and said, Magid, mTOR activity is related to this conjugated triene. So maybe we should leave everything alone and let's focus on the conjugated triene. And that's what we did. So actually, we looked here and we did a lot of Diels-Alder reaction from this big molecule. And we started actually working, and we were intrigued by finding some of the activity showed some of the neuronal survival and doing this in vivo. So Ed went on and he said, very important here if we can do substituted phenyl. We worked in that. All the substituted phenyl were inactive or was not good. Only the unsubstituted phenyl were the active one. But what our intrigue here, you look at here, the double bond. When you do Diels-Alder, the double bond move. So when he reduced this double bond, we found the activity was tremendous. And that's actually was great news for us. We said, okay, let's take that and see if we can have this drug both neuroprotect and neurogen. So here is his summary slide where he actually presented us, and it was intriguing. I just want to share to you with the red part. As I said, we do this color-coded thing. We said not immune suppression. To our surprise, that the one with the double bond did actually show some of the immune suppression. You can see here. So when he did the hydrogenation, there is no immune suppression. And the reason is, this one can do retrodeals go back to rapamycin, which is immune suppressant. So his observation, when you reduce the double bond, you prevent the retrodeals was the key actually to getting this molecule here called ILS 920. And indeed, ILS 920 was great. We show here for neurodegeneration, also for the neuronal survivor. And for the neuroprotection, we were very pleased to see after you do this carotid artery, this is called the middle carotid artery. You get the mouse, you clip the carotid artery, you prevent the blood to go to the brain cell died, and that's your test in animal. Then you do your drug to see if you can protect the brain. But you don't do this for every compound you make. You only do this for selected molecule. And we were pleased to see that this molecule is both neuroprotectant and neurogeneration. We did some affinity labeling study to find out why is neuroregeneration, and indeed we found there is two pieces to that. We found that the molecule binds to another protein called FK binding protein 52. And that's the reason for neurodegeneration. And that actually work was published in the actual PNAS for finding the target. So let me just summarize. Here you took one natural product, and we got 3 drugs out of that. And there are a 4th one, but I can't talk about it, for multiple sclerosis. So here is by doing some molecular modification, semi-synthetic and others, you can actually come up with innovative therapeutic from natural product. Let me take you to the last example here, where we're talking now, many medicinal chemists and pharmaceutical company are worried because company now develop biologic. So as a medicinal chemist, I say, well, I'm not a protein guy, what the heck? But as a medicinal chemist, you can actually capitalize on this and make a small molecule linked to the protein. And that's the area where I want to share with you here in our approaches. So here is when we got the caliche soil sample from Texas, and the chemists worked on that for several years, they were able to isolate this actually cytotoxic agent called calicheamycin. That calicheamycin is so toxic that Phil Hammond, who worked on that, wear double gloves in a separate hood to work because it's extremely toxic. So when we looked at it, we said, we want to find out why it's toxic and how we're going to have a drug out of this. So what we did, and we have George Elstead in my group, George said, well, Magid, I looked into this and I found actually this molecule goes into actual cyclization, and then you come up with the diradical, which take from DNA, abstract the proton, and then DNA will cleave. And that's how cell die, and that's why it's cytotoxic. So that actually was good work by George Elstead, and we realized what's really the initiation for the cytotoxicity of the natural product. So now we have this natural product with all these warhead, because this is how it start, and then we have other component that. How are we going to give this to patient when the chemists working on it wearing all these gloves? So what we thought about, we said, you know what, let's look at cancer and what really some of those actually antigen familiar with some cancer. So we looked at some of those leukemia, and we found that the CD33, and we can generate an antibody, and we can link that antibody to this cytotoxic agent. In order to do that, you have 2 questions. The antibody, where are you going to link it? Antibody have carbohydrate, and it has amino acid, and so on. You link it to carbohydrate or to amino acid. The second question, when you link it and you give it to patient with cancer, Does it reach the cancer cell and it cleave so the toxic material kill the cell? We said, let's find out. And then Phil Hammond went, and Phil actually was a very clever chemist. Phil said, well, I'm going to develop some linker technology. And here is some of the linker technology. After doing some studies, we realized, forget about the carbohydrate, we'll link it to the lysine amino acid, and let's get some hydrolyzable linker. And that's actually the one in blue, was the one of choice. So we linked that to calichamers. And that actually, the molecule here was linked through the sulfur, and then here you have the amino acid actually in the antibody, and that's where you give to the patient as Mylotarg. So that actually molecule will serve you as if the tumor cell, you give the drug, it's like the missile, when you shoot a missile, will look for the tumor cell, go inside, hydrolyze, kill the cell. And that was Mylotarg. And that was actually approved in 2000, and that was the very first example of conjugate drug FDA ever seen. After 10 years, as we saw from Yana's slide today, for the Herceptin, you look at the dates here. So this Mylotarg was 2000, then the company started working on antibody for CD30, or Herceptin, and others, and that shows you how innovation can actually lead to more innovative drugs. Let me finish here and give you just a summary that we tried to talk in early on. What the challenges as a medicinal chemist to make molecules which you know have a good chance of success. And we discussed and we showed some input. Then we took you to some innovative example where we showed you variety of approaches where from natural product, small molecule, and antibody conjugate. As I mentioned today, I was just actually a cheerleader of the group, but the people who did the work are shown here, and I was very pleased that I worked with such talented scientists. Now at Temple University, we have actually substantial effort for cocaine addiction and actually working for drugs which lower the cholesterol is a new innovative mechanism, and also we have drugs working for diabetes. And thank you for your attention and taking the question.